My name is Kevin Kraus, and I spent the last 2 decades in diagnostics, bringing innovations in science and medicine to patients and providers. It's something that I'm passionate about, and I'm excited to talk to you today about what we're doing with PCR, infectious diseases, that we believe can be done at anywhere. So there are 13 million people that die every year from infectious disease, and it's not because of the lack of treatment, but rather because people lack access to affordable and rapid and accessible testing. And so the breakthrough testing is PCR, and that's the gold standard, right? You probably heard about it during COVID, etc. But the thing is that today it's largely confined in the lab. So what if you could take a swab, put it into a dongle on your cell phone, have it analyze that, send the results to your phone, and then send that to your doctor, send that to an online pharmacy? Sounds great, but why haven't we done it? Well, part of the problem is that sample purification is one of the key steps in PCR. And that's how you extract the DNA or RNA. But to do that, you also need to preserve the sample, and those are done in sample media collection devices that have inhibitors. Those are toxic, those are expensive, and they can also affect sometimes the quality of the result. And they're run by skilled clinicians, and they're expensive to do. So what we are going to do here, and what we have, is we have a way to do direct-to-PCR without extraction. So you simply take the swab, put it in our buffer, and use our reagents to analyze and amplify the DNA. And we learned that it's faster, it's cheaper, and it's highly accurate compared to conventional PCR. We started in cancer diagnostics at Colorado University, and we evolved from that, and Summit Biolabs was created during the pandemic when there was a high demand for COVID testing. The company ran over 590,000 samples and generated $14 million in revenue. Now, as the pandemic ended and COVID testing went down, Transformative Biotech was formed and licensed the IP and filed 6 patents, including a lab-on-a-chip device. And we had our first patent issued in July of this year on the chemistry. So we're very excited about the fact that this very simple enabling chemistry is a fundamental thing that anyone doing PCR can use. But we want to take it a step further. And the future we're building is to use advanced microfluidics, a lab-on-a-chip, to take that, miniaturize it, and put it onto a dongle-type device, disposable, that you can then use on a phone. And so you could do it at home, you could do it at work. Maybe you're here not feeling well at this seminar and you want to try and see if you have flu or COVID, right? Or you could do it on a battlefield if you're a soldier, and that could give you timely information. So what we want to do is we want to service this large market, $20 billion. And it's fragmented, I would say, because there's work that's being done in PCR in the research market. There's also sample collection media devices and large incumbents across this. But the thing that they all struggle with is they all use extraction for their primary method to do the PCR. And so we believe that in the molecular diagnostics business, which is the biggest segment, you'll see here that there's ways to save cost and money on those cartridge-based systems. And then also in the point of care, where it's more, there's different applications, including some small startups here that are trying to do sexually transmitted diseases. We think that we can also help enable their devices. And so our target market is about 12 billion of that, and you can see what we think we can capture. And we really believe also that the point of care market is really the fastest growing, and that's where the opportunity is for us to either build our own at-anywhere device or to license and enable third parties to build their own new generation of instrument-free PCR tests. So we have a path to break even, and we have scalable growth. We know from our previous work with the chemistry that we can have high gross margins there. And we also think that there are a lot of licensing opportunities, and we've had discussions with several of the strategics that I showed you before, and there's licensing potential. We're raising $6 million, and that first tranche is about $2.75 million. So far, we've raised from family and friends, And right now what we are doing is we're putting together the designs for the data that we want to get published through some of our partners, and also then build a 510K so we get clearance on the sample collection media. And then we are working to expand our data package and claims across other pathogen and viruses. And we'll work through different licensing and customer options as we bring on new team members. We have a very strong team. I started out many years ago at Ventana Medical Systems. It was acquired by Roche, and then I spent about 15 years there in the US and globally working in different commercial roles, and spent some time also working at Genentech and Third Rock Ventures. And the rest of the team here have experience working not only together at Summit, which was the previous company, but also in large pharma companies, molecular diagnostics, and really have a great sense of urgency and purpose around what we're trying to do to change the world here with our chemistry and our solutions. We have a team who is world experts in microfluidics, engineering, and in microbiology, and we're working and bringing on some other industry advisors as well. We believe that the future really is bringing affordable rapid testing for PCR to patients everywhere. We have the team we think that we can do it with. We have a growing list of advisors. We have our first landmark patent in the US, and we have 5 others pending, including a lab on a chip. And we have the data that we've shown on the clinical testing, over 590,000 samples that really de-risked what we're working on. And we also believe that we can have sustainable growth in the market and also some different exit opportunities among the strategics that I mentioned. So if you're interested in learning more about this, please come talk to me after. We're at booth 58. And I believe that not only can we really grow this business and make it interesting for many different players, but it's also something that we can make a difference and save a lot of lives, because testing is key to knowing what to do for patients who have infectious diseases, not just in the US, but globally. Thank you very much.
You mentioned cost and time, so I was
Yeah.
kind of curious for both.
Sure.
So processing time, and also, what's your target for the dongle?
Yeah, dongle, good question. So it sort of depends across the different products, right? The first one is the sample collection media, and that's a lower cost item, and we think we can shave about I would say 20-30% off of the cost of that. But we have to make our own device. Our goal in the end is, with the handheld dongle, is to get it under $10, maybe under $8, right? And then if you charge $20, $25, then you have a really good margin business. Because today, what's happening in the market is the competition who are making like cart-based systems, like Visby. You may have heard of Visby Medical, and they have an instrument you plug into the wall, and they sell it. They had a special over the holidays for chlamydia gonorrhea for $150 a kit, and that's great, but that's not affordable for the direct-to-patient market, in our opinion, and we think it's also because the cost of that manufacturing is high. So if they could reduce their cost, they're a potential partner where we could use our enabling chemistries and technologies to work with them to get it more accessible out there in the market. Yes, Bruce?
You have 2 slides where you talked about licensing and
Yes.
revenue, and
Yes.
both big numbers. Can you talk about one of those?
Yeah, sure. Would you like me to go back to it or just speak to it?
Just
Yeah.
$20 billion. It's an easy number.
Yeah, 20 billion. Yeah, exactly. So the molecular market is the biggest, right? That's the centralized lab business, and those are the cartridge-based systems. And so the way that we've looked at that market is, okay, how many players, including my former employer, Roche, how many do we have out there, and what's the volume of PCR tests? And our thought is that based on what we've done in the previous company, Summit, we know what the cost of that is when we use this chemistry versus the standard conventional PCR. So let's just say, for example, we could save $2 a test, and there's 2 million tests a year, right? You save $4 million for a partner. Now, they may have to redo some of their studies and their cartridge, let's say, but that's straight to the bottom line. And I know from my past experience, you're always looking for ways to save on the cost, because market share is hard fought, and you need to always be dropping cash to the bottom line from your current install base. So that's the molecular market.
I think that's
Yes.
And to get there, you talked about getting to some cash flow
Yes.
neutrality.
Sure.
That's 4 times your product revenue.
Right. Yeah. So the licensing.
How do you get people to do it?
Yeah. No, that's a really good question. So we have one of the advisors that I'm working with has a lot of experience in licensing, and so our belief is that, and the way we've modeled it is that we're going to be able to go to a number of different strategic partners, and we've had discussions with several, one who's under NDA, that are interested in licensing that technology and then designing it into their kit. So that's really the licensing part. And it could be that we license it to somebody for STIs, or we license it to somebody else for a respiratory panel, or somebody else working in TB. So there's different possibilities there. Yeah.
Is the intention to develop this first and only for individual patient use, or you look at it in terms of staff, in terms of being used by clinicians?
Yeah, the interesting thing is that we don't necessarily want to build a whole portfolio of devices. I want to enable the devices that everybody else has already made out there, and because I know from my experience that if you have to go and get dollars within your roadmap to go fight for changing the chemistry of something that's working, you're not going to do it. But if somebody else outside the company has developed something like that you can integrate into your technology, then that's a win. And so I think we're looking to show that, for example, we can build the point of the handheld device, but we may not eventually be the ones commercializing that. We may just license that out and have other partners help us co-develop that, right?
So what valuation are you raising?
Yeah, so we're raising $6 million, right? And right now we've raised about $855,000 in friends and family. So somewhere in I don't know 3 to 4 million valuation. So we're still early days. We're pre-revenue, but we also believe that because we've de-risked the chemistry and that it has applications, we can customize it per different partner depending on what their targets are, depending on their system. It's a very basic patent that covers swab, saliva, blood, and also other types of sputum, urine. So it's really an interesting start to our IP portfolio. Yeah, thank you. Other questions? Maybe from the audience there.
Yeah, I got time for one more.
Great. Yes, please.
So there's been a lot of money that's spent on very, very low cost.
Yes.
So $5 to $7 a test is a bold claim.
Yes.
Many, many people have made it, and none have even come close to doing it.
Yes.
So what is the secret sauce that allows you to do this? Because that is a huge cost.
Yes.
And as someone who has funded these things through BARDA,
Yes.
government,
Yeah.
how much does it cost?
Yeah, so the individuals at the different universities in Colorado that I mentioned we're working with, they have technologies around the thin film and how to mix it, and also how to use capillary flow. But there's also, perhaps it's a combined combination of that and sonification using ultrasound to disrupt the wall of the DNA or RNA. And it may depend on the target too, right? So that's what we need the money to raise and to, try to figure out, okay, exactly how much time is it going to take and how low can we get the cost of the device. But that's the goal.
Thank you.